All right, so now we're ready to set up the PCR reaction. The PCR reaction is going to use our template DNA of genomic DNA. So remember, this is genomic DNA that was isolated from flies that were ground up. And the two different sample groups that we want to work with are the ARC82 FRT42D chromosome over curly O. Those are our controls. And then the FRT42D ARC82 N14 over curly O. That'll be our experimental. That's where we're looking for that mutation in the EGFR gene. So we are going to only amplify the EGFR gene in order to confirm by sequencing the presence and location of that mutation that showed up by whole genome sequencing from Nevada State College. And so we have primers that we just resuspended. This was our original starting stock. I'm going to put that away in the ice bucket so I don't get it confused. And then we have the working stock that's available for us to spike right into our PCR reactions. So one of the best ways to do this is to make a master mix. And what that master mix includes is everything except for the DNA sample. That will help keep the PCR reaction consistent between the two. So if something went wrong, you know that it wasn't because you forgot to add an ingredient. It was potentially something wrong with that genomic DNA, that it didn't serve as a good template. So I'm going to do that in just one of the two of the PCR tubes that we will use. These are these little tiny baby tubes. I'm going to close them. And I'm going to set up the reaction in just one of them and then take half of it and put it in the other. I'm going to label the tubes on the side so that we know which one's which. One I labeled C for control and the other one N14, which you can't really see. That will have our N14 genomic DNA in it. And I have these tubes as well as everything else on ice just to keep things cold. The master mix that's used for PCR does have DNA polymerase, which is an enzyme, and proteins need to be kept on ice to stay stable. All right, so in order to do this, we're going to take the PCR master mix. It's really great. You can buy these master mixes that already have everything you need in them. So I am going to pipette 50 microliters because I'm doing this for 2 reactions. So getting out my pipette tip, which you can't really see, 50 microliters of this PCR mix. And that is going to go into one of these tubes on ice. Get rid of the pipette tip. And then we need 2.5 microliters of each of the primers per reaction. Well, we have 2 reactions, so that's going to be 5 microliters each, and that will be using that working stock that we just made. So here's the forward primer, 2.5 microliters of that. And here is the reverse primer, 2.5 of each, so 5 microliters total of that. All right, so the next ingredient that we need to add, and the only other ingredient that we need to add, is water. So the final volume of this reaction for each reaction is 50 microliters. So we want to then, by knowing how much genomic DNA will we add to each sample, what that volume should be. So if it's 25 microliters of the PCR master mix, if it is 2 microliters of each of the enzymes, so that's 5 microliters, so now we're at 30 microliters, and we're also going to add 1 microliter of the DNA sample. So that means we only need to add 19 microliters of water, and you want to use sterile water. But since we have 2 reactions, that is 38 microliters. And I'm switching pipettes, by the way, so if I'm reaching up, that's what I'm doing. This is a pipette that's P2, oh sorry, this one is 20 to 200, so that's good to pipette the 38 microliters that I need to do. But before, when I was pipetting 5 microliters, I did that, actually, I didn't use that one. I used the P20, which goes from 2 to 20. So set this to 38 microliters and add our sterile water. Close it right back up, as sterile as possible. and then add that water to the tube. All right, so we have everything added besides the genomic DNA into our one tube here, but we have two. So before we can proceed, I want to mix this really well. So I'm going to go back to this vortexer, mix it up. All right, and now I'm going to take half of that reaction, so that will be 49 microliters because we just have 1 microliter left, and that is our DNA. And that will go into the other tube. We'll add this, put this right back on ice when I'm done. All right, so our master mix is ready and aliquoted into those tubes, so they're ready to go. I will now be able to add the genomic DNA. So the protocol says to add about 100 nanograms per microliter of a concentration of DNA and 1 microliter of that. So I found 2 samples that were around that concentration, and I'm going to add those. Now, since this is only 1 microliter, I am going to use a different pipette, which I have in my research lab, that is called a P10. So this goes up to 10 microliters. And so it more easily is used when you have these smaller volumes like 1 microliter. So go down to 1 microliter. I've already flicked the genomic DNA that I thawed on ice to resuspend it, but it's never a bad idea to do this again. So just to make sure that it's ready. And this is the N14 sample. It's really important that you know which one is which, of course, when you're working with these. N14 is here. That's my tube labeled with N14 on the right. Put this, just lift it into the tube. It's always best to hold the tube so that you see what you're pipetting into. And now I can do that for the ARC82 control. 1 microliter. Again, look to make sure that you're pipetting properly into the tube. Close it up. And then genomic DNA is a little fragile, and you shouldn't really vortex this to shear, because it can shear genomic DNA. So vortexing, as I've been doing, is probably not the best idea. So I'm just going to flick those tubes in order to mix the reaction. Pipetting up and down is another way to mix, but it's probably not the best idea with genomic DNA. These are now ready to go in the thermocycler.